Chapter 2 is about cells and cell division and going through it. When you were in regular biology, we had a chapter about cells, we had a chapter about cell division, and we had a chapter about biochemistry, macromolecules, proteins, carbohydrates, lipids, nucleic acids. So we are going to combine all of those into this one chapter, but a lot of the stuff you guys should have heard of already in regular bio. So, case study, I actually have a video. The first thing it talks about in the book is a boy called Gaucher disease. It is genetic. It means he cannot break down a particular type of fat, which then accumulates in the liver, spleen, and bone marrow. And it mainly affects people of Eastern European Jewish descent. So this is just a little 2-minute video of him and his story, which we'll watch because we'll talk about it later.
I had my first signs at the age of 5 when I started having needle-like, very sharp pain as if like a screwdriver was being pushed through my joint.
Nathan was seen by hematologists, by rheumatologists, even by the infectious diseases doctor. But all the blood tests were coming back normal.
My orthopedic surgeon, after one of my surgeries, told me that I was an enigma.
Year by year, the situation was the same, where he developed another limp, where he developed another excruciating pain.
I did wonder, why can't they solve the problem that I have with my joints? And what are they not getting? I was willing to do anything.
It saddens me that the diagnosis had not been made earlier, but unfortunately, this is not uncommon, especially when patients present atypically. The diagnosis can be significantly delayed.
The result came back with the Gaucher cell identified.
Gaucher disease is a lysosomal storage disorder caused by the deficiency of an enzyme. The liver and spleen may be enlarged in a patient with Gaucher's disease. About 15% of those patients may experience bone pain.
Dr. Pistorius explained that this is the whole reason why I was having all my symptoms. It was the center of it all.
We immediately started him on enzyme replacement therapy, administered once every 2 weeks.
Emotionally, I got better, and physically, I got better. And everything was good. I started to think of Dr. Pistorius as a hero. We're in Dublin, Ireland, in a park somewhere up in the hills. It's a change from New York. It's all kind of miraculous. So I'm walking up to the steps of Dr. Pistorius' office. It's very exciting.
Would you like to see Nathan?
Going up the stairs.
Is Nathan here? Is he?
It's a moment of truth.
Dr. Pistorius will have a surprise for you.
Oh my God.
I'm glad that you did get started on the treatment because it's obviously allowing you to be healthy and pursue your dreams.
I want to become a doctor. I wanted to be to other people what my doctors were to me.
Okay, I'm going to pause it there really quickly because this part's the important part. One of the important parts of the video is doctors often miss the signs of this disease. And it's actually really important to study different diseases of different cultures because this is an Eastern European disease, or Eastern European / Jewish disease. There are diseases that affect Hispanic people more. There are diseases that affect European people more. There are diseases that affect Black people more. So it's important to have a doctor that is well-versed in all of these different cultures and how they can affect all of those different races. So the treatment he mentioned is enzyme replacement therapy. It's 1 to 2 hours of intravenous is IV treatment. That's the liquid that drips into your vein through a needle once every 2 weeks. However, it is extremely expensive. It's $150,000 to $200,000 per year. Another treatment that he could have gotten was a bone marrow transplant. However, a bone marrow transplant would have been a permanent cure, but there is a shortage of bone marrow donors. There's a shortage of blood donors. There's a shortage of organ donors. There's a shortage of bone marrow donors. So because Gaucher disease is livable, you can live with it. The question is, do you make him spend that money on the enzyme replacement therapy? Or do you do the permanent cure when there are more serious diseases that need bone marrow transplants, like leukemia? People with leukemia, that's most of the time a deadly disease, and those people need bone marrow transplants in order to survive. So where do you put the bone marrow? In the people that are going to die, or in the people that it's just a treatment when there's a shortage of it? That's one of those ethical questions that doctors have to ask themselves. So, this we just went over. Which treatment should be covered by insurance too? Our insurance sucks, and our whole insurance system sucks, and it only covers stuff that is absolutely necessary. So, review. This should be familiar to you guys, like I said, from regular bio. We are organic beings. This means we are made of carbon. When you see organic, I know at the store you think it's natural and grown without pesticides. Organic just means made with carbon. You are an organic person. Many carbon-based molecules are made of many small subunits bonded together. Monomers are the individual subunits, and then polymers are the large chains made of the many monomers. There's a little picture where just one individual is the monomer, but you put a bunch of them together. And poly means many. You have many all put together in a chain-like formation. Right. Our cells are constructed from 4 classes of macromolecules. There's carbohydrates, consist of simple sugars called monosaccharides and large polymers called polysaccharides. That's where the mono and the poly are the same. Lipids, fats and oils, phospholipids, and steroids. They're not technically polymers because they're not made of the exact same repeating unit over and over and over again. But they are considered a macromolecule because they are large. Proteins are polymers of amino acids. So amino acid would be the monomer. The polymer is called protein. And then nucleic acids. Nucleic acid would be the polymer, and it's made of monomers called nucleotides. All right, and there's a table. This is in your book and this is in here. I won't go over this table. It's your subclasses, which are your monomers. There's examples and there's functions. Those examples are going to be big in your assignment this week. So bookmark this chart. This chart's going to help you for one of your assignments this week. Cell structure, the basic structural and functional unit in all organisms, including humans, is the cell. And we had a whole chapter on cells in regular bio, so this should be review. Cells differ in size, shape, function, and life cycle, but the basic structure is similar for all cells. All eukaryotic cells, which is what we are eukaryotes, meaning we are made of many cells, have a plasma membrane, cytoplasm, organelles surrounded by their own membranes, and a nucleus that also has its own membrane. Membranes are huge within the cell. And we say eukaryotic as opposed to a prokaryotic cell. To refresh your memory what a prokaryote is, it's like a bacteria and it just has a cell wall and a cell membrane. It has some DNA in the middle, but it's not separated by its own membrane. And then the eukaryotic cell has all these organelles. All right, all cells, both prokaryotic and eukaryotic, have two cellular domains. They have the plasma membrane and the cytoplasm. And cellular domain just means a domain is a place where things happen. So there's a lot of stuff that happens in the cytoplasm. There's a lot of stuff that happens in the cell membrane. The cell membrane just isn't solid, but we'll talk about that in a few minutes. Genetic disorders alter cellular structure or function. Disorders can infect entire cells in the whole body, or a certain set of cells in a particular place in the body. You can have a disease or disorder just in your heart cells, or just in your stomach cells. They can affect a certain organelle in all cells. Gaucher's disease affected the lysosomes, and just the lysosomes of that boy's cells. A plasma membrane. It's also called the cell membrane. You'll see it both ways in various questions. Separates the inside of the cell from the outside environment. It regulates what goes in and out of the cell through embedded molecules. It's like this, it's like our skin. Our skin regulates what comes in and out. And there's little pores in our skin that moisture can either come in or out. And the cell membrane looks like this picture right here. It's one big circle, but this is just a portion of the cell membrane, and they have all these embedded molecules in them for different reasons. The endoplasmic reticulum is a mouthful, so we just say ER for short. It's a system of cytoplasmic membranes arranged into sheets and channels. It's found in the cytoplasm of eukaryotic cells. It's right outside the nucleus. It synthesizes and transports gene products. So this is a little cartoon of it, and it's cut out in a circle because it's right outside the nucleus. The nucleus would be right here. And there's little dots all over it. That's your rough ER with ribosomes. And then your smooth ER doesn't have ribosomes on it. Your rough and your smooth is just about how it looks. It's either studded with ribosomes and looks rough, or it is not, and it looks smooth. And then under an actual microscope, this is what it looks like. And that's a really good microscope. Your average microscope that we have in class can't see that detailed. Your lysosomes and your Golgi apparatus. Your lysosome is a membrane-bound structure containing digestive enzymes that break down old cell parts. And this is what that boy was lacking that had Gaucher's disease. His lysosomes were malfunctioning in all of his cells. So his disease just affected that one organelle in all of his cells. And then the Golgi apparatus is a series of flattened sacs that sorts, packages, and delivers proteins that were made in the ER. It's kind of like the post office or UPS or FedEx. It receives proteins from the ER and it kind of repackages them and distributes them to wherever they need to go. Mitochondria. Singular is mitochondrion, but mostly when we talk about it, we talk about plural, many. These are membrane-bound organelles, are found in the cytoplasm of our cells, and they're the sites of energy production. This is where all of our energy comes from. And there are diseases that just affect mitochondria where people get really, really tired because the place where the energy is supposed to be processed is just not working in them. We have a whole chapter on cellular respiration and how we break down food for energy using the mitochondria. So we'll go into this in greater detail in a later chapter. The nucleus is its own membrane-bound organelle in eukaryotic cells. It contains the chromosomes, which is where our DNA is. It also has the nucleolus. It's the site where ribosomes are made. So the whole thing is the nucleus. You can see the ER right outside of it. And then the little ball in the center, that's the nucleolus. And they sound the same, but they are different organelles and they have different functions. You're gonna have to remember that for your quizzes and tests later this week. The cell cycle. Now that we know what cells are, we're moving on to the cell cycle. The cell is divided into three parts. The cell cycle is divided into three parts, excuse me. Interphase is the time between cell divisions. A cell spends most of its life in interphase and includes three subphases called G1, S, and G2. We'll get to what those mean in a minute. Mitosis is the division of the nucleus and the segregation of the chromosomes into future daughter cells. And then cytokinesis is the division of the cytoplasm. The little GIF is just kind of an overview of mitosis and cytokinesis. Mitosis is the division of the nucleus. Cytokinesis is when the cytoplasm actually splits. So here's the cell cycle overview. So interphase is all of this brownish. So G1, interval of cell growth before DNA replication. The S phase is the interval where DNA replication takes place, where the chromosomes are duplicated. G2 follows DNA replication, cell prepares to divide. So you can think of G1 and G2 as growth phases, like G stands for growth. And then mitosis has the four phases: prophase, metaphase, anaphase, telophase, and then you have cytokinesis where it actually splits. And now there is a phase during G1. Cells can leave the cell cycle and enter an inactive state called G0. This is when cells aren't functioning well. Something's wrong with them. We don't want them to divide. We want them to die. So they enter the G0 phase, they become inactive, and they don't spread whatever's wrong with them into future cells. Chromosomes are the key structures as the cell moves through the cell cycle. A chromatid is one strand of a duplicated chromosome. So on the bottom, you have one chromatid. And then sister chromatids, once they replicate, you have two of them and it looks like a little X structure. Each carries identical genetic information. And they are held together by the little dot at the center. The center of the X is called the centromere. Stages of mitosis. Prophase. The chromosomes become visible. They thicken or condense. Sister chromatids are joined at the centromere. You can see in the little GIF when it starts in a minute. So when it starts, it just looks like little squigglies. And they get thicker, they condense, and then they form the little X structures. Metaphase chromosomes move to the middle of the cell, and the spindle microtubules, which are the little yellow net-looking things attached to the centromeres of the sister chromatids. You can see on each side of the centromere is one of the spindle microtubules. Metaphase lines up in the middle. That one should be easy to remember. Anaphase, the centromere split. The sister chromatids separate. So the net, the spindle microtubules, they pull the chromosomes apart to opposite ends of the cell. And then telophase, chromosomes of the daughter cells decondense. So they once again look like little squigglies. They don't look like the X's anymore. And the nuclei reform. You can see the nucleus reform in the two that are going to be daughter cells. There's your summary of mitosis: prophase, metaphase, anaphase, telophase, and a couple of GIFs of the entire process. So you guys can watch those at your leisure. The green one goes really fast. The one on the right goes a little bit slower. And then cytokinesis. Cyto means cell. Anytime you see the word cyto, it's referring to a cell. There's cytokinesis, cytoplasm, cytoskeleton. Yeah, all have to do with the cell. And then kinesis means split. So a cleavage furrow divides the cytoplasm equally into two daughter cells. Cleavage furrow is just a little point in the middle where they split. And the organelles are distributed to the daughter cells. So each daughter cell should have one nucleus and a set of ER and a set of Golgi and lysosomes. All the organelles split into the two different cells. Mitosis is essential for growth and cell replacement. Some cells continue to divide throughout the life cycle of the organism, while others do not divide in adulthood. So children have a lot more mitosis going on because they're still growing than adults. There's something called the Hayflick limit. Hayflick was a scientist who figured this out. Cells can undergo only a certain or a limited number of cell divisions before they stop dividing. It's genetically controlled. You inherit this skill from your parents. Human embryonic cells, so the embryos, while they're still in utero in their parents or in their mother, divide about 50 times, while human adult cells only divide between 10 and 30 times. It allows growth into adulthood and for cell replacement. Our bodies are continually undergoing mitosis because things like our skin shed. We need to develop new skin cells every day. Our blood and our bone marrow, they go through a lot of mitosis because our bodies are constantly making new blood cells. But things like our kidney or our liver, they don't need to divide all the time. We don't need to replace those cells all the time. They function just fine over a long period of time. Yes, Shay, it's also why you should change your bed sheets at least every week. That's true. Your skin cells shed all the time, and most dust in your house are dead skin cells. You can see when your skin's dry and you kind of go like this on your arm, you can see your dead skin cells kind of come off. If you use lotion, then you don't see that as often, but not everyone uses lotion. Alterations in the control of cell division can lead to genetic disorders such as premature aging and cancer. There's a disease called progeria when young children look like they're 70 or 80 years old. This is a picture. His name's John Tackett. He was 15 years old when this picture was taken, but he looks about 80. He's the oldest known person to survive to that age from that disease. So this is a real disease. It's rare, but it does happen. And there's a problem with the cell cycle in his cells. One of those phases isn't working the right way. Meiosis is the other type of cell division that we need to talk about. So mitosis, our body does all the time to replace our body cells that need replacing. And then meiosis is a process of cell division that produces 4 haploid cells containing only 1 copy of each chromosome. At the end of mitosis, we always have 2 copies of our chromosomes. And there's 2 rounds of cell division instead of just 1. Meiosis 1 reduces the number of chromosomes you have in your cells from, it's called diploid. Diploid means you have 2 full sets of chromosomes in your cells, and it cuts them in half to haploid. And why does our body do this for us? Because your sex organs are the only organs in your body that undergo this process. This process produces eggs and sperm, and these are the cells that make babies. And so we only want to give our babies half of our genetic information. We don't want to double that genetic information every single time. We want to give them half, and the other parent gives them half. So our sex organs cuts it in half for us. Our bodies do this for us. And then meiosis 2 separates sister chromatids. And 4 haploid cells result. We'll go through this process. It'll make more sense as we go through it. So this is an overview of meiosis 1, the first round. And it's similar to mitosis. Nuclear envelope breaks apart. The chromosomes condense and start to form the X-like structures. They line up in the middle, they get pulled apart, and then they separate into 2. And they have numbers after the round: prophase 1, metaphase 1, anaphase 1, telophase 1. And this is meiosis 2. That 2 follows kind of the same kind of steps and turns into 4. You guys have these pictures. And then there's a summary of the entire process: metaphase through telophase 1, and then prophase through telophase 2 and cytokinesis. And the GIFs and overview. The 1 eventually turns into 4. The end result is 4 with half the information from your regular cells. So comparing meiosis and mitosis, like I said, mitosis, you start with 1, you end with 2. And the 2 still have a full set of chromosomes. In meiosis, you start with 1, you end with 4. And the end result has half the information. Has only one set of chromosomes. Random assortment and crossing over. Our genes, our chromosomes, when they split, they are randomly assorted. They could be any of these combinations. And then during prophase 1, homologous chromosomes will pair up, and then crossing over can take place. So the little legs of the chromosomes just switch information. So that when it gets split up and passed down, each one of these, this one is going to be all purple, this one is going to be half purple, half blue, this one's going to be half blue, half purple, this one's going to be all blue. And all 4 of these options are going to have different genetic combinations to potentially pass down to offspring. So meiosis results in 2 kinds of haploid sexual gametes. Like I said, this is where sperm and eggs are made. Sperm is produced by males throughout spermatogenesis is the process in the testes, and then oocytes, that's our fancy scientific name for a girl's eggs. It's produced by females. That process is called oogenesis, happens in the ovaries. Sperm cells called spermatogonia line the tubules of the testes. They divide by mitosis from puberty until death, producing daughter cells called spermatocytes. So men produce sperm their entire lives. Spermatocytes then undergo meiosis, and the 4 haploid cells that result are called spermatids, and they eventually develop into a mature sperm. We won't watch the videos together, but if you want to know more about this process, there's like a 3-minute video on it. And then oocytes are eggs. Females, the production of gametes is called oogenesis and takes place in the ovaries. Cells in the ovary, known as oogonia, begin mitosis and early embryonic development. So they start developing when girls are in the womb, in their mothers. They finish a few weeks later, and that's it. There's no more mitosis. Females are born with all the primary eggs that they will ever have. So girls, we make eggs while we're still in development in our mother's womb. And once we're born, we have all the eggs we're ever going to have, which is a lot of them, but we don't develop more. The guys produce sperm throughout their whole life. We don't do that. And all the primary oocytes begin meiosis during embryonic development, and then they stop. They remain in meiosis 1 until the female undergoes puberty. After puberty, usually 1 primary oocyte per month completes the first meiotic division. The secondary oocyte is released from the ovary and moves into the oviduct. And there's an overview video about this, because I know I'm visual and I need pictures to go along with my words. If you want to learn more about how your eggs are made, or how a girl's eggs are made, you guys can watch the video.